Welcome to this chilly night, both inside and outside. Our speaker this evening is Dr. Alan Kuzirian, and Alan and I were graduate students together, not too many years ago, in which probably we both launched our interest in marine animals about the same time. Alan is now a research scientist at the Marine Biological Laboratory, and tonight he's going to talk about marine biomedical models, white mice from the sea. Alan?
Thank you. It's a pleasure to be here, and I am pleased that so many people braved the cold weather to come out. I'm glad it's cool in here for two reasons. One is I still have my jacket on, and I have to talk, and also I think the tendency is, or the folklore is, that people tend not to fall asleep when it's cooler and not warm and comfortable in a darkened room. I hope to enlighten you a little bit tonight, as well as entertain you and tell you a few things about marine white mice, the biomedical contributions that marine animals have made to natural sciences and to basic biological processes. So if I could have the first slide. One never knows exactly where to begin. So I've given this lecture before to elder hostile groups and at different universities. And so when I always start off introducing where we come from, you know. And so this is the Marine Biological Laboratory, the laboratory down the street. And I always have to tell people that Woods Hole is actually a physical place, right? And there's at least 5 major institutions. There's the Marine Biological Laboratory, and then across the street from the Lilly building, there's the Bigelow building, which belongs to Woods Hole Oceanographic, and then there's National Marine Fisheries, which came first with Spencer Baird in 1871, and then there's U.S. Geological Survey, and there's Sea Educational Associates, and now there's George Woodwell's group, the Woods Hole Research Center. So we always tell people this, and so I don't know whether I should tell you or not, but anyway, I just did, right? And then I always have to allay people's fear and tell people that Woods Hole scientists don't live in a different century. I mean, we don't live in our ivory towers, that we try to keep up with the modern times, and we look just like ordinary people, right? This is actually Whitman, C.L. Whitman, the first director of the Marine Biological Laboratory. And I always tell people that MBL was the first U.S. marine station. It was founded in 1888. It was founded as an outgrowth of Louis Agassiz's school, his summer school, to teach students to study nature and not books, right? The famous Louis Agassiz saying. So, and then I also tell people that our students also keep up with us, right? And we go out and collect like we always do, and go tide pool collecting, and that our research vessels aren't quite as dramatic as the Atlantis, and we don't have ROVs that find Titanics and things, but we're not overcrowded, and we certainly do have plenty of students to fill our research vessels, and we do study nature and not books. And our laboratories are modern facilities, right, as this botany lab is. And people from the 1890s, where these slides all come from, actually, most of them were involved in taxonomy at that point, and trying to figure out how things reproduce, how they fit in the natural environment, and how to classify them. But now, back to our century, I'm going to skip through the phylogenetic ladder and tell you a little bit about the uses, the biomedical uses, the research uses, and the natural biological processes that these animals have been important in elucidating for us. This is a protozoan, one single-celled animal. It's a hypertrich ciliate. The animal's surface is coated with hair cells, or hairs that move and beat, and that moves the animal through the water. And this is actually nature's answer to the bumper car, because when the animal encounters an obstacle and it hits it, it turns around and goes backwards. Why does it do that? It has, in the membrane of this animal, there are channels, ion channels, calcium channels to be more specific, and when calcium enters through those channels, when it hits something, the cilia beat in the opposite direction. The nature, the ionic activation of those calcium channels, the passage of the calcium through those channels happen exactly in this ciliate as happens in nerve cells, it happens in hyperactive cells, secretory cells. The nature of those ion channels have been conserved phylogenetically, and so by studying protozoans and the calcium channels in protozoans, you can apply that nature, the biophysics, the chemical nature of the channel itself, to humans. These are sponges. We'll move up a little bit. And this slide's compliments of Paul Dayton. And Paul Dayton is a marine benthic ecologist. And if anybody knows the works of Paul Dayton, he'll know that this came from the Antarctic. And these aren't ordinary sponges. These are over 6 feet tall and over 3 feet in diameter. These vase sponges in the Antarctic grow to extreme size. But why are they important to us now? The pharmaceutical industry is extremely interested in sponges because of their natural products. Natural products chemistry in the pharmaceutical industry is extremely important. These sponges have been found to contain and to produce anti-inflammatory compounds that are used to treat rheumatoid arthritis, that are used in organ transplants, in grafting operation, skin grafts, to reduce the inflammation to allow the tissue to take. These are natural products that are produced in these sponges and now are being extracted by the pharmaceutical industry. Sponges also have the unique ability of being able to recognize their cells. So cell-cell interactions are also studied in sponges. You can take a sponge, a green one or a red one and a yellow one, and homogenize them up and make a yellow slurry or a red slurry, put them in a dish together, and in a day or two, all the cells will sort themselves out, and you'll have a red sponge again and a yellow sponge. Each cell knows its next nearest neighbor, and they reform in exactly the way they were before you chop them up. So the cell-cell recognition, the process of how cells recognize their neighbor as being their neighbor and not as a foreign substance, is also important in cancer research as well as in the inflammatory conditions in rheumatoid arthritis. So sponges are providing some of the answers to those kinds of questions. This jellyfish is in the phylum Coelenterata or Cnidaria, and it's in the coelenerates that the first nervous system or nerve net system is established. And within the bell of this animal, there are collections of nerve cells that form an interconnecting network, and the ion channels in these jellyfish are a cross between those calcium channels that we found in protozoans and the sodium channels that we find in nerve cells in humans. Peter Anderson at the Whitney Lab is studying jellyfish and their nervous system, and has found that the sodium channels act perfectly good as one would characterize a vertebrate sodium channel in any neuron. However, if you use sodium channel blockers like tetrodotoxin that people use to characterize the nature of those ion channels, they have no effect. If you put them in sodium sea-free water, there's no nerve conduction, so it has to be a sodium channel. But he found that if you put calcium channel blockers on the sodium channels in jellyfish, they block the channel. So there's a phylogenetic transition, his hypothesis is, between the importance and the dominance of calcium channels in lower protozoans and in the development of the sodium channel in the nervous system of higher invertebrates and vertebrates. So the jellyfish allowed his discovery of that phylogenetic progression. And also the ctenophore, the goose sea gooseberries that we see here around Woods Hole very, very often in the spring and all summer. The coordinating beating of these rows of cilia are coordinated, again, by calcium channels and sodium channels, and this provides a very good medium and a model system for studying it, because you can get at the cilia and the channels and the nerve cells that underlie it extremely easily. This, anybody know what this is? Any guess? Well, this is a flatworm from California, Prostes cereus bellostriatus. It even sounds as pretty as the animal looks, right? In the 1930s, when neurobiology was beginning to come of age, these flatworms were used as the first learning and memory models. And in fact, California Biological sells kits for studying learning and memory in flatworms. And when I reviewed the science fair, both at Falmouth High School this year and in Bridgewater at the regional one, there were 2 or 3 projects that I reviewed, I was a judge on, and they were studying learning in the common freshwater flatworm Dugesia that we can get in freshwater ponds. But these animals were very important in the early stages of neurobiology and in the very beginning of the studies of where and how learning and memory takes place. This is a worm, Amphitrite, and these are the gills, and you can see the gills are quite red. And also in this sabellid worm, this is the feeding crown that creates the feeding currents. These are filter feeders. And several years ago, we presented a program at the University of Massachusetts Medical School in Worcester. And we presented talks such as this in explaining biomedical models. And one hematologist was extremely interested in the worms because the red color is due to hemoglobin. Hemoglobin occurs in these worms, and they're the oxygen binding carrier that is responsible for circulating oxygen through the animal's body. And the hematologist at UMass Medical School was interested in other kinds of hemoglobin proteins that might be able to elucidate how some anemias occur and how they can be treated. So these worms, because they have hemoglobin pretty much homologous to our hemoglobin, provided a working model for him to study anemias. This is also a worm, polychaete worm, Myxicula, and it has a neat lifestyle. It builds this nice mucus tube that's usually much further down in the sediment. This is in an aquarium in the lab. But it responds to tactile stimuli and also has very primitive eyes, so it will respond to shadows and things. But it has this incisional axon, very large one, not as large as the squid giant axon, but it's coiled as well. And if you irritate the animal, he retracts very quickly down into the bottom of the tube, and the fast action potential, the movement of the axon or the nerve impulse down the axon in this coiled worm provides a model for the study of nerve conduction in unmyelinated nerves. How does nerve impulses travel very quickly in unmyelinated nerves? It's being studied in Myxicula, and it's actually being grown and cultured at the University of Maine in the Darling Center to use as a learning model for electrophysiologic studies, both at universities and teaching hospitals, as well as at research institutions. These giant barnacles from California and Oregon and Washington coast tell a story about serendipitous collaborations occurring. This giant barnacle was being studied at the University of Washington at Friday Harbor, and Dr. Shimomura, who studies the protein akorin that is bioluminescent in the presence of calcium, he was studying akorin. The people in the next lab were studying muscle contraction in the giant barnacle. And that particular summer, they the muscle physiologists realized that calcium was important in the contraction of the muscle. So they were talking with Dr. Shimomura, and the two collaborating groups got together and injected the protein aquarin, which in the presence of calcium bioluminesces, and that's where the beginning of the calcium imaging studies actually occurred. Totally serendipitously, because somebody was studying muscle contraction in a giant barnacle and the other person was studying bioluminescence. So Dr. Shimomura has been at the MBL for many years. They've been able to extract the gene that produces the protein aquarin. They can splice it into other animals. We're making transgenic zebrafish, and so wherever the precursor for the protein occurs on the gene and in the division of the cells, whenever calcium enters the cell, it lights up. So calcium homeostasis, the importance of calcium as a second messenger, is now being studied visually by the luminescence of the cell, by having the protein aquarin incorporated into the gene structure of that cell line. And it all started with a barnacle and a jellyfish. This box crab, Lithoides, is important to both behavioral and neurobiological studies. The animal has behind the two eyes here two glands called the green glands that produce neurohormones. Serotonin is the principal one. Serotonin controls behavior. Everybody knows about Prozac now, right? Controlling behavior and adjusting serotonin levels. Well, crabs produce serotonin. It modulates their behavior, where the crabs and also lobsters are aggressive or passive. It also controls whether their molting cycle, and also now the hormone, another different hormone in that gland, the growth hormone, also resides there. And an investigator, MBL investigator at the University of Connecticut, has been able to extract that growth hormone and now uses it in the shrimp culture industry to grow larger shrimp so that we can enjoy them at Christmas time or whenever. So all started with neurobiological studies and neurohormone studies in crabs and crustaceans, right? Like this one. Everybody knows what this one, certainly it's a gustatory delight. But how does the animal find out and locate its mate and its food in the water supply? Yellow atma at MBL and at the University of our Boston University Marine Program studies how these animals smell in the water. Chemotaxis and chemoreception. And the animals have also been used as a model now to create a lobster robot. The neural circuitry involved in olfaction of the lobster has been defined rather clearly, and the people in MIT, working in conjunction with Yella, have created a robot, a lobster robot, that senses, not smells now at this point, but changes in salinity and orients to the odor plume or to the salinity plume according to the strength of the signal that it receives in the sensors that's on the robot. And the object is to use the robot to locate pollution plumes in aquatic conditions. So here we have olfaction in a lobster being used in the neural circuit that controls the animal's behavior to create a robot who will help us find sources of pollution in aquatic environments. Rather neat and unique example of biological application and science transfer, technology transfer. Limulus polyphemus, the famous horseshoe crab, the living relic, developed or evolved for about 350 million years. It has been responsible for Nobel Prize to two MBL researchers, Kiefer Hartline and George Wald, studying photobiology. There's a pair of eyes here There's another set of eyes here, and there's a set of eyes underneath, and I think there's actually 8 sets altogether. But studying phototransduction, how is light energy transformed by the retina of these animals into a nerve impulse? How does that nerve impulse travel through the optic nerve and get imprinted on the brain? A lot of the research that George Wall and Kiefer Hartline did using Limulus was responsible for their Nobel Prize. Now, of course, it's also important in the pharmaceutical industry because within its blood system, it has a protein called Limulus lysate, and this lysate coagulates in the presence of the toxin produced by gram-negative bacteria. Strep and Staph and Fred Bang and Stan Watson were the ones that discovered it here in Woods Hole, and associates of Woods Hole came out of that. And so now in the pharmaceutical industry, all injectables and pharmaceuticals that are in liquid form and that can't necessarily be autoclaved is studied or tested for sterility using the Limulus lysate test for purity. This is the blue mussel with a nudibranch on it, but the byssus threads are what's important in this slide. Glue, natural products, glues in barnacles and glues in the byssus threads of mussels, natural cements. And there's a company that produces Cell-Tac. Cell-Tac is a natural adhesive that we use in cell culture, that's used in orthopedic surgery and prosthesis construction. All came from analyzing the structure of the natural protein, both in the blue mussel and in the barnacle. The surf clam, Spatula. We all know it if we eat Snow's clam chowder or order clam strips at the restaurant, right? Well, biologically, it's important to a whole group of MBL summer scientists who come to study its dividing cells, the gametes. The animal is so large that the gametes make up a good almost 2/3 of the portion of the inside of the animal. You can collect up to 30 to 40 milliliters of gametes, eggs or sperm. The animals have separate sexes. You can mix the sperm with the egg in the test tube, and you have so much material, dividing material, the divisions are synchronous, that you can study how the chemical processes of how cell divides. Cyclins, which control cell division, were discovered using Spatula, and they're dividing cells, and now you can actually make and homogenate without having the cells present at all, and you can recreate the spindle in a test tube without any kinds of cell membranes present at all. So cell cyclins and the process of cell division is being studied with surf clams, because if you know how normal cells divide, maybe we can discover why cancer cells are dividing differently. So there's a lot of cancer research money going into supporting research on surf clams. The Woods Hole squid, very important in the early days of the MBL. The process of nerve conduction was discovered or rediscovered in the giant axon of the squid, and Hodgkin and Huxley got their Nobel Prize in 1963 by studying and coming every summer and studying the Woods Hole squid. The cephalopods are also important in learning and memory, because the octopus and the nautiloids are used in learning and memory studies. They have an extremely, and this is sepia, the cuttlefish, they have extremely good visual acuity and use patterning to signal each other, both as warning patterns. You have mate selection patterns. And so what is the ability of this animal to use visual cues in its learning and memory is being studied at MBL. And Roger Hanlon, as the new director of the Marine Resources Center, is a cephalopod biologist. he's interested in reproductive behavior as well as learning and memory studies. So these cephalopods have been important in discerning the limits and the processes of learning and memory in these animals, and it's directly related to that process in humans. Starfish, this or the phylum Echinodermata, the sand dollars, and our regular, very familiar starfish were important in the early days of the MBL in the study of embryology, how cells divide and which cells, tracing the fate, the cell lineages of starfish and echinoderms were extremely important in knowing which cells formed which tissues, which formed which organs. So in the early embryological studies, marine invertebrates, especially the echinoderms, were extremely important in ascertaining that knowledge, and they're still used today in the process of cell division, both in normal cell division as well as in cancer cell division. The protochordate, the acorn worms, and the urochordate, the sea squirts, the ascidians, are important phylogenetically because their larvae contain a notochord, and that notochord is formed by neural ectoderm, and that neural ectoderm that forms the notochord in sea squirts and protochordates is the same process by which our nerve cord and vertebrate and backbone are formed. So these groups are the transition groups between the vertebrates and the invertebrates, and their immune system is also important because they're used to study AIDS in the process of anti-inflammatory processes as well as autoimmune diseases. Now, I'm going to tell you a little bit about some of the research that I've done, and the epistobranch mollusks is what I classify myself as a malacologist who studies mollusks, and in particular the epistobranchs and the nudibranchs, and they are rather colorful animals of the sea. They come in certainly rather bizarre shapes and colors. Some of them are camouflaged themselves within their natural environment. This is sargassum weed, and this is the nudibranch scilia. Others have very distinct warning patterns, and this purple one from California, Flavolina iodinea, with bright orange serrata, bright red rhinophores for chemoreception, and a beautifully purple lavender body. They're certainly much easier and much nicer to study than the neurobiologists who study crickets and other insects. I much prefer to do my research on epistobranchs. But why are they important? This is Navinax inermis. It's a notospidian. It's not quite as advanced as the nudibranchs, but they're used in learning and memory studies because they're used for ingestive conditioning and how the nerve system processes those kinds of stimuli. Also, the C paraplegia are used in both associative and non-associative learning paradigms. And the animal that I work with is Hermesenda crassicornis, and I think both of us are working on it. There we go. They're used as learning and memory models. And why are they good models? Because they're rather small, easy to keep. Their nervous system is extremely small and easily accessible, and the number of nerve cells is much, much less, about 10,000 cells in the nervous system of Hermesenda as opposed to 3 or 4 billion in the human brain, and half that or so in the mice or the rat. And they are capable of behavioral modification with very distinct learning paradigms, and also they can be kept in laboratory culture as white mice are kept in culture. So here's the nervous system or the brain of Hermesenda. It has a cerebral pleural ganglion and some petals. What's important to us in the discussion is the eye, which is located here, and the statocyst or vestibular organ, which are located here. These cells, there's only 5 cells in this eye, and there's only 13 cells in the vestibular organ. So you can see it's much more simple to get at this animal, to get at these nerve cells, and the fact that these nerve cells are very constant in their position. You can study the same cell between animal and animal and get the same results. You can inject the cells with fluorescent dyes, and you can follow the alburization in the synaptic endings of these cells. So you can see that the eye with the photoreceptor and the hair cell in the vestibular organ meet here and synaptically interact. You can put microelectrodes in this cell and in this cell and measure the synaptic interaction electrically between the two cells. You do that enough time and you can create a wiring diagram. So here's the eye. It has two types of cells of the 5. There are 2 A's and 3 B's. The B's are kind of analogous to our rods in our eye. They're spontaneously active in the dark. They're sensitive to very dim light. The A cells are more sensitive to bright light, more analogous to the cones in our own eye. The optic ganglion is a group of interneurons that interact with or in between the eye and the vestibular organ. And again, there's only 13 cells in that organ. We train the animal first. In the early days, we would put the animals in a plastic tube and put the tubes on a turntable. And actually, the first turntable was a Garage 95 record changer. It spun at RPM of 78. So not a long playing 33, but a 78. Anyway, the animal is normally photopositive. It moves to a spot of light. So you can put the animal at the end of the tube and have some photo cells here and shine a spot of light into the center and measure how long it takes the animal to move from the end of the dim light into the bright light, and that's the animal's positive response to the stimulus of light. If you take and present light specifically paired with the rotation of the turntable, you decrease the animal's phototactic behavior. You depress it, you shut it off, and that shut off of inhibition of the phototactic behavior lasts for at least 3 to 5 days. And so there's the measure of learning that only happens when the two stimuli are explicitly paired. Electrophysiologically, you can measure changes in the ionic conductance, and what happens in the paired condition, the potassium channels are shut off, and so the inside of the photoreceptor in the eye stays depolarized much longer and is facilitated. So the next stimulus that comes when they're paired reinforces the biophysical changes that occur in the eye. Part of the learning process involves the phosphorylation of a particular protein. This 20,000 molecular weight protein only appears in animals that have had positive training. So here is another measurement, a biochemical measurement of how and when learning takes place. And now, instead of putting them on turntable, we put them in a plexiglass box, and the box is attached to a stand which is attached to the orbital shaker. And so instead of spinning them, we shake them, which is much more analogous to wave action to which they're naturally exposed. And with Pavlov's dogs, Pavlov trains his dogs with food presented at the same time that a bell is rung, and then when he rings the bell, the dogs salivate in anticipation of the food. Well, with Hermesenda, if you pair the light with the agitation, and then on the 4th day after training, you turn the light on and the animal's foot contracts the same way the dogs would salivate. So there's associative learning and a positive measure of it with Hermesenda. And so that's another indication of the positive training model that we've developed for Hermesenda. And what we've done is to measure the aberration volume where the two systems, the photic system and the vestibular system, interact. And what we find is that there's a decrease, a focusing of the synaptic area of the photoreceptors onto the hair cells in animals that have learned with paired training and rotation, as shown here. But also what was neat, we didn't expect at all, you can distinguish fast learners from slow learners. So you have smart nudibranchs and dumb nudibranchs the same way. We have all kinds of spectrum of learning ability. In humans, we also found the same thing. Unbeknownst to us, in the nudibranchs. This Hermesenda model is important if it's called, or what we call, a high connectivity model. If you can take the information that we learned in the nudibranchs and apply it to vertebrate systems and eventually to humans. So then there is an associative training model in the rabbit. Instead of using light and rotation, they use a puff of air onto the eye coupled with a tone. And you train the rabbit by presenting the tone in the air puff together for a couple of days, and then you test the animal by just placing or just turning on the tone, and the rabbit blinks. And if you look in the hippocampus of the animals that have been trained that way, you find that in the CA1 pyramidal cell region of the hippocampus, which mediates learning and memory in vertebrates, the same kinds of biophysical changes that we saw in Hermesenda, the change in the ionic currents, the change in the second messenger proteins that we found in Hermesenda, they are now finding in the hippocampus CA1 pyramidal cells. So the model is a high connectivity model with Hermesenda because it has direct application to the biophysical changes that occur in learning and memory in vertebrates. Another nice thing about Hermesenda and nudibranchs is they can be cultured. The animals are hermaphroditic. The two animals have to come together, though, and exchange sperm, and then within about 24 hours, the animal begins to lay an egg mass. There's about 1.6 million eggs in that egg strand. And the animals develop inside the egg for about 7 days and then hatch as larvae, and you can see a very nice shell here. So they really are mollusks. They do have a shell at some point in their life cycle. And 30 days later, if you feed them microalgae, they look like this. They have a foot, and the shell is 3 times the size as it was when it hatched. And if you put these larvae in the presence of the adult food, the animal metamorphoses, crawls out of the shell, and begins to take up its adult form. And in about another 30 to 45 days, the animal looks just like the adult, so you can go from egg to egg in 90 days, which is an extremely short reproductive cycle. And if you can keep animals in culture, you can study the neurodevelopment of the nervous system. The first organ, sensory organ, to develop in the nudibranch is the vestibular organ, which is why it was a good model system to choose in the first place. It occurs in embryonic day 4, and then 3 weeks later, the eye begins to form. And how does the eye and its neurons connect up to the statocyst that develops 3 weeks later is a very good neurological question that we haven't answered yet, but we are about to, or in the process of. Here's the eye, there's the lens, the statocyst is outside the plane of the section, but here's the nervous system developing. And if you can study the process of normal development, you can study the process of nerve defects and birth defects. Here's the eye in this particular nudibranch is way up here. The statocyst organ is way down here, and it still has its nerve connection to the right place. Another thing we found in our embryologic studies of nudibranchs and epistobranchs is that normally the shell calcifies in seawater, no problem, but, and this is what it looks like in the scanning EM picture, But if you deplete the seawater of strontium, there's no calcification. It was a new discovery, discovered again serendipitously by Joe Bidwell here at Woods Hole Oceanographic, studying the development of aplysia and the significance of the strontium in mineralization, biomineralization is very important, both to the animal and to where it occurs in estuaries, because we found that bivalves themselves respond to strontium levels the same way that the nudibranchs do. But we're also rather curious that there are 8 parts per million concentration of strontium in seawater. And there was a study done in the 50s of municipal water supplies in 2 Midwest towns. And they studied the incidence of caries in people's teeth. And they found, believe it or not, that in municipal water supplies that had about 8 parts per million strontium, the incidence, the number of people with cavities in cavities was extremely low. And if it went to either side, the incidence of cavities went up. So what is the phylogenetic significance of this? We haven't really discovered it, but that's something for someone to perk in their curiosity. We also developed squid in strontium-free seawater, and this is the stone that activates the vestibular system. This is with 8 parts per million strontium. This is a squid that was grown in seawater without strontium, and it's a way of developing atactic animals, which is important because everybody recognize this picture? Right, Neurolab. The Neurolab just came down. There were 4 toadfish in space, and they were studying the animal's adaptation to weightlessness, because Steve Heistein at MBL has developed a technique of surgically opening the animal's skull, taking the vestibular nerve, cutting it in half. Because the animal's powers of regeneration, that nerve will regrow, but he puts in place in the middle of it a wafer plate with microelectrodes that come out, and so as the nerve reconnects through the pores in this grid plate, they're able to do extracellular recordings from the animal's inner ear to see how it adapted in space. So there were 4 of these animals on the Neurolab flight that just came back down, and they were able to study how these animals adapt to weightlessness. Obviously important to astronauts' physiologic conditioning to weightlessness, but also important to vestibular, in studying vestibular diseases like Meniere's disease. When people develop vertigo, how do you treat it? How does it originate in the first place? Well, perhaps with squid and with nudibranchs grown in strontium-free seawater, we can see how they adapt to weightlessness, not in space, but right here on Earth. So that's that's the tact that one could use to study, to use Hermesenda and other epistobranchs with that strontium need to develop atactic animals. So what am I doing now? We tried to take the learning model of Hermesenda, because it is so well defined, and study the effects of lead toxicity. In fact, Tracy Crago handed me a flyer just before the seminar tonight from the Radio and Television Broadcasters Foundation, all about lead learning deficiencies in children. Lead is still a prominent health problem in children, both the exposure from lead paint and the dust that has lead paint in it. Certainly the problem of lead exposure has been reduced because of gasoline. Lead levels have been, or lead has been eliminated from gasoline. But learning deficiency in children exposed to lead, for the most part, is irreversible. And even though the normal lead levels, which were over 30 parts, 30 parts per million lead levels, blood lead levels in children before 1978, when they outlawed leaded gasoline, has fallen to about 3 parts per billion, which is much better. There's still about 100,000 children who have extreme lead poisoning each year. Over 1 million have blood lead levels over 12 parts per billion, and it's at 10 parts per billion where you begin to see lead learning deficiencies. And the problem with that is that at 10 and 12 and maybe up to 15 parts per billion, you don't see those learning deficiencies until the kids get to be about 8 or 10 years old. And by then it's too late, because the deficiencies are organic in nature and it's irreversible. So we thought that in working with Herman Epstein, who had done studies with learning and lead in children and also in mice, that, wow, we have a very nice system in Hermesenda. The learning is well defined as a very simple nervous system. What happens if we expose Hermesenda to lead? Well, the first thing we noticed was that the animals respond by stopping feeding in a very dose-dependent manner. Anything above 3 parts per million exposure of Hermesenda to lead shuts off learning or feeding. What I do know from past experience is that serotonin, the neurohormone serotonin, modulates feeding, the feeding program of Hermesenda. Lead is known to affect neurotransmitters and neurohormones. We tested the animals by training them in the usual way, and we found that animals kept in natural seawater worked or learned quite well. Those exposed to lead had definite learning deficiencies. We took and cultured the neurons from Hermesenda nervous system. Shown here is a normal neuron growing in culture medium. We exposed the neurons to very low lead levels and watched what happened. The neurites began to degenerate very, very quickly. Lipofuscin bodies occur within the cell soma very rapidly, and the cell dies within about 2 or 3 days, depending on the level of lead. There's been a new study that says that lead triggers apoptosis, programmed cell death. And basically what we've seen in our lead culture experiments is apoptosis, the induction of programmed cell death. So one way that lead may work in neurotoxicity is to cause early apoptosis, and that's why the learning deficiencies are irreversible in children, and hypothesis that we're studying. We can take and put the eye in cell culture, and we can measure directly the effects of lead on the biophysics and the physiology of the photoreceptors of Hermesenda's eye. And I'm in the process of writing a grant to study the effects of lead, and the basic hypothesis is that lead affects the neurohormones that modulate learning and memory in Hermesenda. So I can't tell you the answer to that. If the grant gets funded, maybe in a year we can come back and do it again. But anyway, because Hermesenda is such a useful model system for studying learning and memory, the parameters are so well defined, it does present a good model for studying lead toxicity. So I've hoped that these examples have given you an idea about the new white mice and what we've been able to learn from them. So as you walk along the coast and the seashore and behold the beauty of nature, that you stop perhaps and look down at the tide pool at your feet, but look through the reflection and wonder at perhaps the secrets of nature that lie within the animals of the new white mice of the sea. Thank you very much. Questions? I've overwhelmed people. Yes?
You were talking about the gluing effect of the barnacles and that.
Uh-huh.
This last week I had my mooring weight and I was trying to clean it off for reconditioning for next year.
Right.
And oh my goodness, I had to use sandpaper on it and it was just, whatever they hold on with it, it's amazing.
It's a protein. The exact nature of barnacle cement, to my knowledge, hasn't been entirely worked out yet. Was it Tom White that was here at Hui, was the first one to define chemically the Bissus cement, and it's from his work here at Hui, and then he moved to Maryland and formed his own company that produces cell tack, and cell tack is what we use to glue neurons in cell culture to the cover slip so that we can do these experiments. And one thing that I didn't show you a slide of, but we kind of came across as we were doing the cell culture. Hermescent is difficult. Aplysia is much easier to work with, both in culture, whole animals, and in neuronal culture. You can just put aplysia neurons in seawater and add some glucose to give it some energy, and they grow very well. But with hermescent, you need a very defined medium. You have to be very careful how you separate the cells. So we would have to physically digest away a little bit of the protein, the intercellular cement, and loosen up the neurons. But then we had to physically tease apart the neurons and put them in cell culture. And we had coated the cover slips with cell tack, but as we used the little fine needles to dissociate the cells, we'd scratch the cell tack. What we found was the cell bodies like to sit on the cell tack, but the neurites that grow out like to follow the groove in the cell tack that we made with the pin. So it essentially create a track. So what we'd like to try is to take two cells like the photoreceptors from the eye and the hair cells from the statocyst, make a track between the two and watch the neurites grow along the track. And then we have seen neurites crossing each other and touching each other. We haven't done the EM to show that they actually synaptically interacted. But we have a great potential then of using cell tack grooves to take and create mini neural networks. And then you can study all kinds of things because you know how those networks function in the animal. Then you can recreate or change the parameters in cell culture. So you have a very plastic system that has great potential.
That happens to be a fiberglass board too.
Yeah, well, it glues, and it does it not just by hydrogen bonding, which is what most glues do. They just hydrogen bond or static bond between the chemicals that make up the two components that you're trying to glue together. But these adhesives actually integrate much more tightly, and not just by hydrogen bonding. So that's why they stick so well. Yes, Allison.
Did the toadfish survive their different states?
Yes.
Do you know anything about the results?
I don't know anything about the results. I do know that, like the day or two before the shuttle was to return, that all four were still alive. But I haven't spoken with Roger Hanlon. Was in communication with Steve and Steve's postdoc over a culture grant for toadfish. And so Steve was really, really pleased and amazed that all four of them were alive, and they were getting recordings from all four fish, which was really kind of neat. This thing started out about 5 years ago. NASA held a conference here at MBL, and we were invited to come and to discuss possibilities of which animals to take in space. And everything is regimented so tightly. Everything has to weigh as the minimal amount possible. Life support system has to be as simple as possible. The experiments have to be able to be done by the astronauts with minimal training. And so this was one that worked. The Japanese had already created an aquatic life support system. We just had to modify it a little bit to take salt water because they had done it for goldfish. And so the support system worked very well. The electrodes coming out of the fish's head were put into a harness, and so the astronauts just had to take their plug from their computer and just stick it in basically into the plexiglass box, and they were able to do the recordings. But the technology that goes into creating a, quote, a simple experiment in space is never really simple. So they were very pleased that with all the effort that it took to get them there, that they were able to get some recordings done. Yes?
What are the extractions
from the sponges? They're steroids. They're steroid compounds, natural steroid compounds that are anti-inflammatory, and they've proven very, very effective in treating rheumatoid arthritis. And the exact chemical nature of these steroid compounds, I haven't read enough to give you much more information than that. But they are beginning to study them chemically and being able to reproduce them synthetically and use them as in clinical trials and as prescription medications.
So what is the work a lot on sponges?
Yes. Right. And he also, that's right, and cell recognition and the messengers, arachidonic acid and how that's involved in cell recognition. Yeah, he's been doing that for many years. All done at MBL as well. Yes?
Well, the research on the surf plant is really all about the fact that they've done some completely new inventions or whatever on the mice, which they say is really quite, it's a good result that they just kind of start doing it on people.
Oh, the angiotensin and angiostatin. That's a little bit different. The whole process of angiotensin and angiostatin is based on the fact that some or many tumors that grow in very contained fashion, right, respond or need a very large amount of blood in order for them to grow. And they produce angiotensin, which creates new blood, very tiny capillaries that keep the tumor growing. Angiotensin and angiostatin prevent the formation of that capillary network, that blood supply, and therefore the tumors don't grow. But that doesn't say why the cells became cancerous in the first place. So the research that uses the surf clam and what controls cell division, normally and both atypically, will still need to go on, because one is just creating the angiotensin angiostatin basically creates an environment where the tumor can't grow and survive. But what we don't know is what caused the cancer cells to become cancerous in the first place. Why is their cell division non-stoppable, basically? And so what people are studying with the surf clam is the normal process of cell division and hopefully looking for changes that, like cell cyclins, were not known until about 4 or 5 years ago. And now there's cell cyclin 1, 2, 3, I think there are 5 different cell cyclins that are involved as second messengers to setting up in initiating cell division. So changes, genetic changes or mutations in the genes that cause any one of those cyclins may be responsible for cancer and why cells begin to divide uncontrollably. So the basic research into the mechanism of cell division will still have to go on. Clinically, you can treat the tumor and make it disappear, and again, it depends on what kind of tumors there are, because there are some tumors, like ascites tumors, that don't form a nodule, that just actually just slough off cancer cells into a fluid medium, and therefore angiotensin angiostatin would be totally ineffective on that kind of tumor. It's only the very contained ones that need the blood supply to keep going and getting larger where it will be effective. But some kinds of cancers it won't touch at all, because the nature of the growth of the tumor is entirely different and doesn't depend upon creating a new blood supply in order for it to keep growing. Yes?
The hair cells and the statuses.
Uh-huh. Yeah.
You said that strontium kept them from forming. Is that stopping, like, otoliths forming?
Yes, that's right.
The hair cells themselves still form, it's just they don't form the otoliths.
Right, right. Yeah. The statuses, both in the human ear, the inner ear, and in these very simple statuses, the calcareous stone brush against the cilia. The cilia are what we call kinocilia. They beat at a particular frequency. As it interacts with the stone, the beating frequency slows down, and also the pressure distorts the cell membrane. That opens channels, and that's what creates the nerve impulse. So if you don't have the stone there to interact with the cilia, then you have no impulse generated. It's not quite that clean because the human system also responds to angular acceleration. So there's fluid in the inner ear which does move, and so you get some sensation, but of angular movement, but the actual gravitational force always down, right? That's mostly caused by the interaction of the calcareous stones with the cilia. So that kind of, quote, weightlessness is missing in these animals. So, and that's what happens in space too. Yes?
Is the regeneration capability of the toadfish unique?
No, it's actually many lower vertebrates and certainly all invertebrates are what we call multipotent in that they have very good powers of regeneration, and which allows then these kinds of studies to be done. Hopefully, there has been and continues to be a lot of research in limb bud regeneration in things like salamanders and primitive vertebrates, so that injuries, spinal cord injuries, and nerve cord or major nerve injuries can be studied so that perhaps they can also be regenerated and recreated. What was unique is in Heistein's work was that the vestibular nerve is easy to get at, and that the animal does have the powers of regeneration. And what he does is cut the nerve and put in a wafer grid, and the nerve grows through the holes in the grid, and that's allows him to do extracellular recordings as the hair cells fire and respond to gravity or lack of it. Then you can do the recording. So it's just basically using the regenerative power of the toadfish to the benefit of being able to get the nerve to grow through the grid, and you can attach the wires, and then you can study it remotely without having to have the fish held steady and to do everything while the nerve is exposed, basically. Yes. Oh, John. Good question. I don't know the answer to that, and I'm not sure that people do. It's related to which genes turn on at which point. It has to do with advancement in the continued. It's like Occam's razor. You get more and more specialized. Therefore, you lose the ability to go backwards and to become a generalist. And so, as the genetic machinery becomes more and more defined, it loses the ability to go back the other way. And so, that's as much as I can tell you with certainty at this point. Otherwise, I'll get into an area and get into trouble. Because I haven't kept up, and things change so rapidly that you can't keep up with everything. So I figure I do reasonably well to keep up with the neurobiological literature. We're now getting into lead, so I have to read all that environmental toxicology stuff, and to remember my marine invertebrate stuff, because I haven't lectured and taught marine invertebrates for a long time. The head's getting tired, so I'm going to stop while I'm ahead. I haven't kept up, and so basically it's related, I think, to the genetic machinery. The more and more it gets defined, the less ability it has to go backwards, and it's where the activators and the promoter genes are, they can become more and more restricted, so therefore you can't go back the other way. All right. Thank you very much.
Audience participation event, Sounds from the Sea. I hope it'll be a fun interactive event. Michael Moore, Doug Millichek, for both the biology department, Joe Warren, who's a graduate student in the U.S. Engineering program, and Carolyn Levi from the National Aquarium, who will be playing tapes with sounds and recording from the sea and challenging the audience to come on stage. So it should be a fun evening. Tuesday, 7 o'clock.